When I say that I study bacteria and viruses, people usually assume that I study different kinds of disease. And in almost every conversation, there is a point where the person talking to me gets really confused when I try to explain that I study how bacteria avoid getting sick. I'll explain. All organisms get sick sometimes, and the most common cause of disease are viruses. They infect humans, they infect plants, they even infect bacteria. Viruses that infect bacteria are called bacteriophages, or just phages for short. And no, this is not something out of a sci-fi movie. This is what it looks like when phages attach to a bacteria cell and infect it. After infection, the phages replicate inside the bacteria cell and eventually kill it. So viruses really do cause disease in all forms of life. In fact, they have been causing disease since the beginning of life. 4 billion years ago, when life on Earth had just appeared, viruses were already there. So to survive, life had to develop defense mechanisms against them. Today, all living forms of life have immune defenses against viruses. Our immune system is incredibly complex. To defend ourselves from disease and prevent infection, it requires coordination and communication between various different cells. However, almost every cell in your body can sense that it was infected by a pathogen and has different tools and mechanisms to try and stop the infection. For a long time, we thought that our cellular mechanisms were unique to animals because we couldn't find them anywhere else. However, in recent years, research is finding that more and more of our cellular mechanisms actually also exist in bacteria. The conservation of cellular immune mechanisms between human and bacteria indicate that our immune system evolved a lot earlier than we thought. I'll focus on one very important mechanism. When our cells sense that they were infected with a pathogen, a series of events take place that end up with the cleavage of a protein called gasdermin. The cleaved gasdermins bind together and create these ring forms that penetrate into the membrane of the cell and lead to the death of the infected cell. At the same time, alert signals are being sent out to the rest of the body. This protects us in two ways. One, if the infected cell dies before the pathogen can replicate inside of it, it prevents the spread of the infection. And two, those alert signals recruit specialized cells of the immune system to the site of infection that can use other mechanisms to get rid of what's left of the pathogen. For a long time, we thought this was unique to animals, until my advisor, Dr. Tanita Vine, had discovered that bacteria can do the same thing. And I don't mean it conceptually, I mean that they have the exact same proteins. She discovered that when bacteria are infected with phages, the same processes take place, gasdermin proteins are cleaved and create these pores that lead to the death of the infected cell. This protects bacteria because bacteria replicate by duplication and live in colonies of identical cells. So if infected cells die before the pathogen can replicate, it prevents the spread of the infection to the entire population. However, one question still remains unsolved, and that is whether bacteria also send out alert signals to each other. This raises a fundamental question in evolution because immune signaling and communication is one of the hallmarks of immunity of complex organisms. If it exists in bacteria, it could indicate that it evolved before complex organisms even existed, which means that immune signaling and communication evolved in parallel to the evolution of complex life, of multicellular organisms. How did these two processes affect each other? One way to try to answer these questions is to study how conserved defense mechanisms operate in organisms that are kind of in between, like multicellular bacteria. I usually get very confused faces when I say this. I study a very unique type of bacteria called streptomyces that are special because when they replicate by duplication, they don't detach from one another. They create these long strings of attached cells that have very complex structures and have very unique features. For example, they have division of labor. Some cells are in charge of attaching to the surface and others are in charge of replication. And they're also known to have a lot of signaling molecules that help them coordinate these processes between them. We were able to find that they have genes that encode for gasdermins, or that we predict encode for gasdermins. In fact, it seems that gasdermins are very common in Streptomyces species. This tree shows gasdermin proteins from a variety of different Streptomyces species. And I don't only predict that they have the gasdermins, but also that they have associated with them the necessary genes to activate the entire process, as well as proteins that could either produce signaling molecules or sense them. So in my research, I study how gasdermin systems defend against viral infections in Streptomyces species. But more importantly, I study how they help them communicate with each other, and how this communication helps the entire bacterial population to avoid disease. So by studying how conserved immune mechanisms operate in organisms that vary in their complexity, I hope to find another missing link in the story of how life evolved from unicellular defense to multicellular immunity. And with that, I would like to thank all of you for listening, but also a special thanks to my advisor, Tana, my lab mates, and all the people who support me in my research, but also to all the incredible women who made this event possible today.